Hey guys, Mr. Vree again. We're going to do a quick lecture on mutations here. By the end of this, I'd like you to know the different kinds of mutations and some things that can cause mutations. So a couple things to keep in mind is that mutations can happen in two different spots during DNA replication or during transcription. Though we have proofreading systems, they can still occur. Lots of examples of this in popular society, like in movies, Teenage Mutant Ninja Turtles, X-Men, and so on. But they can also happen in real life, as you know, and they can produce a variety of problems, and in some cases, mutations are not always bad. They can be good too. Before we get too far, one thing that I need you to remember is that if you have a mutation that occurs in your gametes, then that is going to be passed on to your children. And so if that's something that you were born with, then that's something that you will likely pass on to your children. But if that mutation happens in your body cells after you're born, let's say your skin, that's unlikely that it would be passed on to your children because those cells would not be in your reproductive cells. The first kind we're going to look at are point mutations. And so point mutations occur when there's a single base change in the DNA. In some cases, it doesn't make any, it will result in a change in the protein. And so if you look at this fictitious example, if our original DNA said the dog bit the cat, and we change it to the dog bit the car, if we have a one base change, it can mean that it means something very different, and so that's just an example. And so if we look at a real example with this top line being the DNA, the second line being the messenger RNA, and then this being the amino acid sequence, you can see right here if the DNA changes from a C to a T, then our messenger RNA will produce it, instead of a G, an A. And so in this example, instead of having glycine be produced by the GGG codon, our codon now reads AGG and we get serine. And so that could produce a different protein. So we would call this type of point mutation a missense mutation, or it doesn't make the same sense that it made before. However, some point mutations can be silent, and this is a good example right here to help you wrap your brain around this. So if we said the dog bit the cat with a C, and we change it to the dog bit the cat with a K, you can see that it still pretty much says the same thing, and so it doesn't have an effect on the organism in this case, or in the sentence, and what it means. So in this real example with bases, you can see our DNA sequence right here reads CCC, and in this one it reads CCA. So you can see the codon would be GGG, and in this case it would be GGU. So this is a good example. If you were to check your codon chart, you can see that that third letter in our codon chart oftentimes codes for the same exact amino acid. We call this the wobble effect. And so that same tRNA can carry glycine if it's GGG or if it's GGU. And the point is here that this would be a silent mutation. You wouldn't even know that you have a mutation even though your DNA is different than what you had before, or what your parents inherited or gave you. Okay, so those are the two types of point mutations. Now we're going to move on to a new kind called frame shift mutations. And in a frame shift, we're going to either insert or delete one or possibly even more bases. And what that's going to do is it's going to shift all of the codons one way or the other, and it's going to oftentimes result in a protein that's non-functional or very, very different in how it works. An example of this would be Tay-Sachs disease, which we learned about earlier in the year. So in this example, we're going to go from the dog bit the cat, and we're going to add a D right here, And so now this O is going to shift over, and that's going to be the third letter in our DNA. And so the G is going to move over to a new codon. And so it doesn't make any sense, right? So everything after that point is going to shift to the right, and it's not going to make sense. Here's another example, but this time we're going to do a deletion, and we're going to delete the D, and so that's gone. So now the O is going to shift to the left, and now it says the og bit it heck ats instead of the dog bit the cat. If we're to look at real DNA sequences here, if we delete this A right here, now this C is going to shift over, and that's going to be the third letter in that codon. And so here is that C shifting over, and so now you can see every amino acid is going to be different after this point. And so you can see the dramatic effect that frame shift mutations can have on a protein. So those are the two types of mutations that I want you to be familiar with: point mutations and frame shift mutations. And point mutations can be either silent or missense, and frame shift mutations are the result of insertions or deletions. Real quick on some different causes of mutations. The types of things that cause mutations are called mutagens, and so those could be things like radiation, like UV light, gamma rays, X-rays. They could also be chemical mutagens, like exposure to chemicals or other forms of radiation, chemicals that you find in smoking, and things like that. In conclusion, I want you to remember that, all of life likely came as a result of mutations, and and they're sometimes good, but they can also they're probably usually bad, but they are a source of genetic variation in species. But when they are bad, they can lead to genetic disorders. And so here is one quick example of one that is bad. It's a result of a point mutation. It's sickle cell. And so we've learned a little bit about this, I think, earlier in the year, where we have normal red blood cells here, but when they get a mutation, their hemoglobin proteins, it causes them to have a sickle shape. And so that can lead to problems, clogging of the arteries and veins. As you can see here, they'll accumulate and cause tremendous pain and sometimes organ failures. Here is the actual example. So if we look at the DNA of someone normal hemoglobin, they have a T here on this codon, and if we change that to an A, you can see that the codon and the messenger RNA will go from become an A, or was an A, it becomes a U. And so instead of coding for glutamic acid, I think it's called, it's going to code for valine instead. And so that is what's going to cause the hemoglobin to have a deformed shape. I hope this was helpful. You have the remainder of the hour to work on your reading guides or notes from chapter 14, and tomorrow be prepared to carry out a modeling lab on transcription, translation, and mutations. Have a great day.